progress, so now we're at perform DNA repair. just saying. All right, so here I'm showing you our most, can everybody hear me in the back? Yes? Okay. So here I am showing you our most current understanding of the phylogenetic tree of life with bacteria, eukarya, archaea. And I hope I'm not insulting anyone's intelligence here by just sort of reminding you how to read a phylogenetic tree, because I will show you another one in the course of this talk. And so, the nodes in the phylogenetic tree, the places where branches connect, we consider those inferred or hypothesized ancestors, and we can say that a group is monophyletic if it shares a unique common ancestor unto itself, exclusive, not including other groups. So here, bacteria forming a monophyletic group. Now, the inferred ancestor that I want to talk to you about today, and some people have already brought this up in their talks, I want to talk to you about the LUCA, the last universal common ancestor of all life on Earth, and lived on Earth 3 to 4 billion years ago. And this group probably doesn't need it, but to sort of put that into a pictographic perspective, if we read this picture from left to right, we see early Earth on the left and modern times, including astronomers, people like some of you in this room, on the right. And of course, I'm talking about, do I have a pointer? Oh, sorry. Okay, yes. And of course, I'm talking about a time period right in here. Now, I personally believe, the topic of this particular symposium is, the Dark Ages. And I believe that the LUCA has great capacity to teach us about modern biodiversity, and also some capacity to help us look back further in time. Probably not something I need to convince folks of in this room. And so one way we can understand the LUCA is by studying its genome content, and that helps us to understand what its metabolic capabilities were and what its structures were like. And there have been many studies on the LUCA genome, and I'm showing you some of those here. And the genes inferred to be present in the LUCA based on these studies have been aggregated into a database called Lucapedia. Yes. So, one of the things that fascinates me is trying to understand how did the LUCA conduct its DNA processes? How did the LUCA interact with its own DNA genome? And so, based on these studies of the LUCA, we know quite a bit about its ability to do DNA transcription, or at least a little bit about its ability to do DNA transcription, depending on who you ask, and its ability to do DNA replication. Now, let's remember that DNA replication is not just the ability of an organism to make more DNA. DNA replication requires quite a lot of fidelity in terms of making that copy, and so there's a lot of excision and repair that goes on during that process. So it's no surprise that at least some of the genes that we infer to be present in the LUCA genome were involved in DNA replication, and specifically in excision and repair during DNA replication. And so I became very interested more broadly in DNA repair enzymes that might have been present in the LUCA. And so using Lucapedia and studies in Lucapedia that predicted, using Lucapedia and looking at genes that were predicted to be present by at least 4 studies within Lucapedia, and considering the function of these gene families predicted to be present again by 4 studies, using a gene ontology resource, I asked, how many of these genes, were involved in DNA repair? And out of 981 genes predicted present in the LUCA by at least 4 studies, we determined that 14 of those gene protein families were probably involved in DNA repair. And so what were they doing? Well, some, again, were involved in DNA replication, but some were probably acting outside of that DNA replication system, some operating in meiosis and doing various excision repair. So we decided to look in depth at a protein family called MGMT that's ubiquitous across the tree of life, and therefore we assumed probably present in the LUCA. So here I'm showing you a representative MGMT protein. Now in E. coli, MGMT has 3 paralogs, and they are OGT, ADA, and ATL. The OGT and ADA paralogs are pretty similar to one another. They have an N-terminal domain that does de-alkylation of DNA phosphates, so essentially removing methyl groups from DNA phosphates, a form of DNA damage, repairing that. And a C-terminal domain, so these are bifunctional, a C-terminal domain that demethylates or deethylates O6 guanine or O4 thymine. OGT and ADA act as monomers. That is, they don't recruit other proteins to be part of a complex to do these DNA repairs. They act on their own. And we would say that they're pseudo-enzymes. Why are they pseudo-enzymes? Well, after they do their job, they demethylate or deethylate something, they're defunct. Now, they differ in terms of OGT is constitutively produced, so it's always on, whereas ADA is auto-induced, and in fact, it becomes its own transcription factor. Now, ATL is a little bit different. It lacks this N-terminal domain function, and it has the C-terminal domain function. It is able to bind DNA, but it doesn't actually do the demethylation or do the deethylation. Instead, it recruits other DNA repair enzymes that are not part of this protein family, so unrelated to this protein family, to actually do the DNA repair. And this, we believe, is a true enzyme, meaning it can be reused, and in terms of its production, we don't know if it's always on or if it's induced. So why do we care so much about de-alkylation or demethylation, deethylation of DNA, and why might early life have cared? So thinking in terms of MGMT, so remembering that it is going to, one of its functions is to demethylate or deethylate a thymine or a guanine. So these spaces become methylated or ethylated, and in fact that causes, oh, there was supposed to be a little animation there, I apologize for its absence, but in fact what that causes is mismatch pairing. So T normally binds with A, that's not news to anybody here, but in fact when this is methylated it bonds with G. Similarly when G is methylated it bonds with T, and these point mutations in DNA caused by this methylation or ethylation can lead to, this can be perpetuated through DNA replication or through transcription. So what are the sources of the methyl groups that cause this damage to DNA? Well, they can be endogenous or exogenous. Most often in modern life, they're exogenous sources coming from the environment. However, endogenous sources can include this S-adenosylmethionine. And so those of you that are familiar with this molecule know that we really need it. It's a really important molecule, and it does a lot of good work in the body. However, it turns out that this can, in fact, randomly methylate parts of the DNA, transfer a methyl group to parts of the DNA that we don't want it to do that to, and so actually causing damage. And so one possibility is that as there was more methane present in the early atmosphere, so here showing you early atmosphere of Earth, more methane present, and possibly membranes of early organisms were a bit leaky. We know that this is a very old molecule predating the LUCA, so it's possible that there was more of this floating around in living systems anyway, methylating DNA. Of course, it's also possible that there were other sources for methylation as well. And again, you know, these are repair enzymes that we need in our own systems, and of course, we're not exposed to this level of methane in the atmosphere. All right. So anyway, to convince ourselves that this MGMT protein family was present in the last universal common ancestor, we reconstructed a phylogeny using sampling from across the tree of life, or rather from across the prokaryotic tree of life. Eukaryotes probably would not have contributed very much to our understanding here. I'm sorry to say, because I studied eukaryotes before this. And so, nevertheless, using all three of the paralogs, OGT, ADA, and ATL. And so what we determined is that, in fact, this protein family, MGMT, was present in the last universal common ancestor, this node here representing the LUCA. And following it diversified into an archaeal and bacterial lineage following the species tree of life, or consistent with the species tree of life. And it turns out that the OGT type enzyme was probably ancestral here. So remember, that's the one that's constitutively produced and bifunctional. So then We have the origin of ADA, so again, that one that operates as its own transcription factor arrives later within the bacterial lineage. And ATL, which again is recruiting other proteins from other repair DNA repair families, arise here later in bacteria. I should point out that this ATL is primarily found in bacteria, so probably arising due to a horizontal gene transfer from archaea. So what do these things look like? Well, so these images that I'm showing you here, and I'll start these movies up here in just a second, these are the MGMT paralogs based on crystalline structures shown here from E. coli. And you can see I'm showing you here in light colors. These are the N-terminal domains that are doing the demethylation of the phosphate in DNA. And these darker colors here, these are the C-terminal domains that are responsible for demethylating or deethylating guanine or thymine. And so, just get this started for you so you can see the structures. And to point out here in this ATL, again, as I've already said, we see only this dark colored structure. This is the homologous with the C-terminal domain over here. So only, that function that bonds DNA and recruits other enzymes to make repairs. So how does there we go. All right, so how does the LUCA compare? So we did some sequence reconstruction here, ancestral sequence reconstruction, as well as 3D modeling. And so here's what we get. And what you can see is that there is the darker color, the darker orange here represents something that's homologous with the C-terminal domain that is presumably performing repair by removing an ethyl group or a methyl group from guanine or thymine. And then we have kind of a simple N-terminal domain here, so not quite as complex as what's going on over here at OGT or ADA, so lacking these beta sheets, but still possibly some functionality here in the N-terminal domain. All right, so where are we? Well, we came into the study knowing that LUCA possessed enzymes that acted on DNA, so it was interacting with its own DNA genome. And we know that these enzymes included enzymes for DNA repair, especially within replication. So what we found was that there are potentially more DNA enzymes present in the LUCA than we realized, and not all of those are acting within the DNA repair system, most likely, such as MGMT. MGMT in the LUCA appears to have functions similar to OGT ADA. So, having both an N-terminal and a C-terminal domain, possibly, again, dual functional, like modern MGMT. And the MGMT protein we saw later, or present in the LUCA, later diversified in both archaea and bacteria to produce some additional functionality. So I would go on to speculate that these at least relatively simple DNA repair was probably possible in pre-LUCA organisms. So something that maybe some of you who are studying such organisms should be on the lookout for. And that's all I have to say about those things.
So we have time for one short question.
Very illuminating. I was interested in the final hypotheses, and one of the things, the null hypotheses in your final statement would be that in order to get LUCA, or in order to get the last common universal ancestor, or the unity of biochemistry, you might have had polymerases that made a lot of mistakes, very similar to what you actually have in let's say, viral DNA, because that would allow you more options to arrive finally at the final unity of biochemistry. So it's just another way of looking at it.
No, I really appreciate that comment, and that's certainly something that I would like to think about and integrate into to continue thinking about these topics. Thank you.
Okay, let's thank AJ one more time.